I'm Chantal, a PhD student within the UTS Climate Change Cluster. My research focuses on bacterial chemotaxis, which is a type of behavior that allows bacteria to move toward chemicals that they like and away from those that they don't. It's similar to how we're drawn to pleasant smell and instinctively avoid unpleasant one, except bacteria don't have noses. Chemotaxis research has broad application, spanning medical science and human health to environmental science. Pathogens use it to infect hosts, plants or animals like oyster and coral, while bacteria in the ocean use it to recycle nutrients, which ultimately control ocean productivity. The bacteria I study belong to the Vibrio genus, some of which are big players in ocean ecology, while others are notorious human pathogens. For instance, Vibrio cholerae cause cholera, a disease responsible for thousands of deaths worldwide each year. Recent research in our group has revealed a peculiar behavior in some Vibrio species. They produce a chemical cue that repels their own kind, as if they had a built-in social distancing mechanism. While bacterial attraction is well studied, repulsion remains largely unexplored and has often been overlooked in chemotactics research. We hypothesized that this repellent cue acts as a critical signal, allowing bacteria to disperse in the environment to avoid niche competition. This could be a strategic way for bacteria to expand their range, explore new environments, and access alternative food sources, ultimately reducing intra-species competition and increasing the survival in diverse ecosystems. My aim is to better understand chemotactic repulsion in marine pathogens, ultimately to determine whether self-repellent could be used as a tool to protect economically valuable species like coral, oyster, and even fishes from pathogenic infection. To explore this, I use a 3-inlet microfluidic chip. This chip allows me to measure the chemotactic behavior of bacteria when they are exposed to different chemical gradients. Solution can be injected through each of the 3 inlets into the main channel. These 3 solutions then flow in parallel within the main channel, which is wider. In this case, the bacteria are injected in the middle inlet, while the repellent is injected on the bottom and negative control on the top. The bacteria swimming behavior is recorded and then analyzed to assess the effect and strength of the cues. In cases where repellent cue is introduced, we observe the bacteria actively moving away from it. This research could provide new insight into microbial interaction and dispersal in natural ecosystems, helping us understanding how marine pathogens spread, locate new hosts, and minimize competition. In the long run, this could enhance our ability to predict, monitor, and manage marine disease, benefiting conservation efforts, aquaculture, and human health industries.